Take it away.
Gives me a chance to leave the meeting now. I won't do that. Okay. All right. Well, good morning, everyone, and thanks for the invitation to speak. As Justin said, you know, we go back a ways. I didn't realize it was so far. I'd like to take you know, credit for his, you know, smart insights and find somebody else to blame for everything he does that isn't so smart. But I can't take credit for that. So I'm just happy to have a surgeon interested in these questions. Today I'm going to talk about sensory plasticity and spinal cord injury from bench to clinical considerations. And I'll try to split the talk about 50-50. So the fundamental situation that I think we're faced with in spinal cord injury is shown by this diagram of a hemispinal cord with descending and sensory input meeting in the spinal cord, and then the output of motor or autonomic function. And as you can see, the spinal cord injury disrupts the descending feedback, but also the ascending, the descending input, but also the ascending feedback. But there is probably some degree of remaining descending input and ascending feedback, even in cases that are clinically complete, so-called physiologically discomplete. We don't have great measures of the residual ascending feedback or the effect of what's going on in the spinal cord or what's coming in on the sensory loop has on this ascending feedback and therefore descending input. But that's sort of a subtler point for later. Of course, the big question is what happens in this circuitry after part of it is changed. In stroke, we use the term diaschisis to talk about changes in uninjured parts of the neural circuitry affected by a remote stroke. And we can think about that a little bit here insofar as that the sensory input to the spinal cord below the level of lesion is essentially uninjured. But the big question is it in fact really normal? And between altered descending input, perhaps changed sensory input, altered spinal integration, we certainly will likely have a new motor output or autonomic output. And we already know this clinically because we have both decreased movement, but the addition of spasticity, and we have decreased sensation, but the perception of neuropathic pain. And we have autonomic dysreflexia as an altered autonomic output as well. So the animal model that we're using is a nociceptive reflex, the cutaneous trunci muscle reflex. And the way this is elicited is by a noxious stimulus to the skin causes a little sheet of muscle under the skin to contract. And it's somatotopically organized very well so that where the noxious stimulus is provided is where the contraction occurs. And we don't have exactly this reflex, but it was a very useful reflex for us to study because we have some idea about the wiring diagram. And while it's not simple, it's simpler than most things we have in the spinal cord. So instead of pinching the skin, we can stimulate dorsal cutaneous nerves all the way up and down the spinal column. They are segmental and come into the spinal cord at every level. And instead of watching the muscle contract, we can actually record from the motor nerve. And so we stimulate and record and get these traces over here on the right. The pathway in the middle is a little bit murky. We know there are these propriospinal neurons that are ascending because the motor pool for the cutaneous trunci muscle is at the cervical thoracic junction, and yet there's input all the way down to upper lumbar levels into this circuit. We know that, we know that the shortest latency between the onset of stimulus and a response is short enough that just three synapses between an afferent, two synapses, three neurons, an afferent, an interneuron, and a motor neuron. But there's many parts of this reflex that are much further out and are probably polysynaptic in nature. If you stimulate at 0.5 milliamps and activate only A fibers, you get this early component of the reflex. And if you stimulate at 5 milliamps and activate both A and C fibers, you get both this early and this late part of the reflex. And of course, we can quantify the size of this reflex. So we have quantifiable physiology, and we can control how strong we stimulate and which afferent populations we activate and record the effect of that. Now, the nice thing about these dorsocutaneous nerves is that you can put a spinal cord injury in the middle of the spinal cord, maybe a T9 thoracic injury, and study what goes on with the interrupted or partially interrupted pathways from below the level of injury, but also whether there's any changes above the level of injury. We initially assumed that this would be an unchanged within animal control, and it turns out we were wrong. So if we stimulate either at 1 hertz for 20 stimuli, 2 hertz for 40, 5 hertz for 100, or 10 hertz for 200 stimuli, and sequentially add up these neurograms, what you can see is, depending on the level, whether we're studying things at T10 or T7 input, T10 input, or T13 input, we get a spectrum. That early component is easily seen at all levels. It's a little smaller at 10 hertz than it is at the lower frequencies. The mid-range, this late component is more prevalently seen with 2 hertz and 5 hertz stimulation. So most everything for the rest of the talk will be looking at 5 hertz stimulation. But you can see there's a little bit of time before it takes several stimuli before it grows to its final size. This is wind up and consistent with C fiber physiology, and it tends to fade out at higher stimulation rates as well. But the point is that this can be quantified both in terms of the frequency of stimulation and with the location of the stimulation of input.
And again, Keith, what we're mapping there is that motor, that cutaneous motor response?
Yeah, this is the CTM motor neurogram, and this is just showing them sequentially, 20 of them plotted one after the other. So,
perfect.
So imagine looking down on this waveform and its colors, and that's what each of these rows are. 20 rows here, 100 rows here. Okay? Now, it turns out we can stimulate on the contralateral side and activate this CTM motor nucleus as well. And you can see that the response is less from the contralateral side than it is from the ipsilateral side. But the point is that we can get to this motor nucleus from either ipsilateral or contralateral sensory input. And there's a slightly greater delay to the onset of that first response, which suggests to us that there's another neuron in the circuit at least. And we found no dorsal root ganglion that projected the contralateral side. And as you'll see in the next slide, where we do a hemisection of the spinal cord above here, we can still get the information up to the CTM motor nucleus. So there must be some interneurons that project across the midline to join this pathway to get this reflex to work from the contralateral side. Now I set up all that to show you the quantifiable physiology. We also have done some quantifiable anatomy to sort out why we might have different things going on at different levels. This is a micrograph of the dorsocutaneous fascicle, and this is it blown up to see heavily myelinated, more lightly myelinated, and all the unmyelinated C fibers here. And if you look over here on the right, there's about 10-fold more C fibers than there are A delta fibers, and about 5-fold more A delta fibers than there are A alpha and beta fibers. But there isn't a huge difference across these different dorsocutaneous nerves. It's not like one nerve is bigger than the other in a particularly meaningful way. So we're talking about the same number of afferents, despite the fact that they're in different anatomical locations. But this is really, should give you an idea about how many more C fibers we have than A delta fibers. And most of these cutaneous nerves have a lot more pain fibers than they have large diameter afferents in them. Now we can label, by injecting the peripheral nerve, different populations of afferents from these dorsocutaneous nerves. We can use IB4 to mark non-peptidergic C fibers, and cholera-toxin subunit B to mark A fibers. And what you can see over here is where they project the C fibers here at T7 and then at T13, and then also the CTB, where they project more deeply. If we erase all the other cells and just show you where these terminals are, you can see above and below the dorsal root entry zone at T7, there's relatively superficial projections of the C fibers in red and deeper projections of the A fibers. If you go down to T13, things look somewhat similarly, although the C fibers seem to be grouped a little bit more around the dorsal root entry zone, whereas the A fibers are a little bit more spread out. But we can quantify the amount of staining, the area that covers the staining. And in fact, we can even, looking at these stains, if we co-label for synaptophysin, we can determine how many of those little branches are actually terminal synapses. And, obviously, if you were like slicing through the branch of a tree, you might cut through some twigs, but you might also, cut through a twig where it's giving rise to a leaf. And you can think of that as, so about between 30 and 40% of these terminals co-label and have synapses. So we can even quantify not only the number of sprouts, but the number of leaves or number of synapses that are associated with those additional sprouts. Because if you grew new neurites and didn't make new synapses, it might not make much of a difference. So synapse number matters as well.
And when you're labeling this, Keith, are you finding a specific sensory nerve that you're cutting end on and applying this to, or are you injecting the skin?
We are injecting the dorsal cutaneous nerve directly. Now, the synaptophysin is just applied to the slice to look for those proteins in the central projections. But the labeling of the central projections of these sensory afferents came from injecting the peripheral nerve.
Okay.
With either IB4 or CTB.
Okay.
Okay, so, we decided that we wanted a circuit to study, but we also wanted to be able to justify why to study it. And I'm only going to tell you about the neuropathic pain side of the story. The autonomic story would take another talk at some point. But as we teach medical students, there's a normal response between stimulus intensity and the perception of pain intensity. And it's sort of a sigmoidal curve. There's a point at which, the pin feels sharp, it doesn't really hurt, and then it starts hurting. And then at some point, no matter how hard you poke the person, it's still 10 out of 10 pain. And after spinal cord injury, and often with peripheral nerve injuries and some thalamic strokes, there's a shift in this curve after injury, such that what was previously felt to be mechanical and non-painful can be perceived as painful, allodynia, or things that were mildly noxious are now perceived as being very noxious, hyperalgesia. And so we wanted to know if there's anything that we could learn from this neural circuit about the development of these things, at least at the spinal level. If you want to get really technical, you can't do pain research unless you can ask the person about it. Right. The rats, you can't really tell you on a scale of 1 to 10, but we can at least know about whether there's any change in the spinal reflexes that I just showed you. So we would say if we got anything, it would be nociceptive hyperreflexia as sort of a model for at least the spinal components of what might lead to neuropathic pain. So here's the experiment. We do a right T10 lateral hemisection, and we can label at T7 and T13, both above and below this injury on both the injured and uninjured side. And we can stimulate all the dorsal cutaneous nerves on both sides and record this neurogram and see what happens. Now, we did this injury and then let the animals go 6 weeks. This is the size of the reflexes before the injury. I showed you this a few slides back, but 6 weeks after that right T10 hemisection injury, this is what those reflexes look like. So below the hemisection, there is now increased reflexes compared to when we had an uninjured case over here. But interestingly, there's also increased reflexes above this injury on this side compared to over here. Below the injury on the uninjured side, the reflex has gotten larger compared to over here. And even above the injury on the uninjured side, so the reflexes changed everywhere. There's global nociceptive hyperreflexia. And if we wanted to quantify that, these are the percentages by which things have increased either for the early component in the left, upper left triangle and the late component in the lower right triangle. So there's huge increases in these reflexes physiologically after this injury. And it's not just something below the level of injury. So when some of my patients tell me they hurt all over, I have to consider the possibility that something about their injury has generated a more global change in their nervous system than just things below the level of the injury. It also says that the uninjured side and above the injury can't really serve as a control because they are changed relative to baseline. Now, if we look at what happened anatomically, this is the projection patterns before injury. This is the projection pattern at T13 below the level of injury, and you can see there's marked increase in the projections of A fibers and C fibers. But even above the injury, there is also the same sprouting. So where we have nociceptive hyperreflexia, we have nociceptive sprouting. And when we've done the analysis, this also corresponds with an increase in synapses. It's still about the same 30 to 40% of those projections end up making synapses. So we have quite a greater input And the nice thing is that the physiology and the anatomy co-vary, and this gives them a better chance of somehow being causal, that more sprouts leads to greater hyperreflexia.
And this is ipsilateral and contralateral.
When we looked at these, you couldn't tell whether the T7 was from the ipsilateral or the contralateral side.
Okay.
And that was helpful because both sides were hyperreflexic.
Okay.
So we started asking ourselves, what could possibly be causing this change, 6 weeks out from injury? And so with a collaborator, we were looking at microglia in the spinal cord, marked here in the dorsal horn in box B with IBA1. And in a normal spinal cord, there's only a few active microglia. 6 weeks after this hemisection injury, there's this persistent increase in microglial activation and numbers of microglia. And this is 6 weeks after injury. So all this stuff with, astrocytes and starting to set up the wound barrier is one thing. This is also 3 spinal segments away from the injury. So diffusely throughout the neuroaxis after spinal cord injury, there's persistent inflammation. Now, my colleague was working with a TNF inhibitor called XPro, and this modifies microglial activity. And if we do the hemisection and provide XPro every 3 days for the 6 weeks, then we see that there's no more microglial activation really than there was at baseline. And you can see that's quantified down here for both T7 and T13 on the side of injury, and on the uninjured side, you see these increases in microglia with injury, and then bringing those numbers back down with injury plus treatment. So we said, okay, if we've modified the microglia back to normal, what happens to the reflex? So here's the hyperreflexia of injury alone. If we look at animals that got injury and then 6 weeks of XPro, their reflexes look like they're back down to normal. These are the decreases over here relative to the injury alone situation. So if we compare the non-injured to the injured plus treatment, above the level of injury, it's basically we're back to normal, and the microglia numbers have gone back down to normal as well. So if you control the microglia, you get normalization of the reflex. Interestingly, below the level of reflex, we had the treatment caused there to be a reduction in microglia below the level of baseline of uninjured. And in fact, that generated an even lower reflex, sort of a hyporeflexia below the level of injury. So again, sort of evidence that the change in the microglia does have an impact on the change in the reflex. So then of course we asked, well, what's going on with the anatomy? Here's that sprouting that comes with the hyperreflexia. And after the injury plus 6 weeks of treatment, now we have much less sprouting. We sort of precluded it. So we have less reflex, normalization of the reflex, and largely normalization of the central projection. So over here is the increases in percentage from baseline for injury alone. And with injury plus treatment, we sort of knock it back down. There's still a bit more C fibers at T7, but not as much as when there was injury alone. So we can sort of preclude the sprouting and the development of hyperreflexia by modifying the inflammatory environment over the 6 weeks from the time of injury. So this has some implications clinically that maybe we should be looking at treating the inflammatory response early on, and perhaps we can block sprouting of pain fibers and the development of nociceptive hyperreflexia, and in humans, perhaps neuropathic pain. The issue, of course, is that the only thing that we would be blocking in terms of sprouting or attempts to reorganize things in the nervous system? So I think I've pretty much just said this, that if we were to reduce chronic inflammation, we might get away with, we normalize the anatomical plasticity and the physiological plasticity. And this should be something we start thinking about early on. We do a lot of care in the ICU that is oriented towards keeping the person alive and getting them out of the ICU, and don't spend a lot of time thinking about the neuroplasticity and the rehabilitation that may be needed to be considered rather early in the case of some of these neuroplastic changes. I'll tell you anecdotally that most of these changes are probably going on very early. In our autonomic model, we only give a 2-week survival time, and all the pathology of sprouting and changes in autonomic function are there. So it's occurring relatively rapidly after injury.
Was that a one-time dosing, or were you giving it serially?
X-Pro has to be given every 3 days
Okay.
subcutaneously. Other reports in mice show that it didn't have much of an effect unless you gave it intrathecally, but we can get this clearly with intra-abdominal injections.
And how long do you apply it then? For the full 6 weeks?
For the full 6 weeks is all we've tested. Of course, we would love to know in future experiments, how late can you start and how short can you give it and still have the benefit. It might be something you could turn off early on and not have to dose that long. We just don't know yet.
Right, okay.
So in summary, this part of the talk, we are looking at the fact that we have anatomical and physiological plasticity after injury that, that is problematic, and we may not be losing, we may not just be losing sensory motor function, but we may also be gaining positive phenomena such as spasticity, neuropathic pain, and autonomic dysfunction. And while I didn't show you the autonomic data, it's pretty clear that we're going to have to not only consider intervening relatively early to try to modify this, but we want to get rid of abnormal plasticity or maladaptive plasticity that might lead to neuropathic pain, but not necessarily minimizing neuroplasticity that leads to adaptive physiology that improves function. So we may have a hard time sorting out, if the processes are the same, sprouting and synapse formation for neurological recovery, how to get rid of the unwanted stuff without simultaneously getting rid of the stuff we do want. So that's sort of the story with regards to the preclinical stuff. And so I've shown you back to this diagram again, because we now have to acknowledge that these uninjured sensory fibers may not be normal. We certainly know the pain fibers change. Whether this happens for large diameter afferents is less clear. We need to do the same studies looking at muscle afferents. But at least we know that some of this uninjured sensory input is no longer normal. So we have both a decreased amount of descending input and ascending feedback and an altered anatomy and physiology of the uninjured inputs to the spinal cord, which guarantees that there's going to be some altered spinal integration and a change in motor output. But the flip side is to say, well, this route into the spinal cord is still present. It is uninjured even if it's changed, and we have access to it. And so what can we do, acknowledging that it may not be the same as it was in the uninjured situation, and there may be variability across patients about how much it's changed? Can we still make use of this input to the spinal cord to drive some better output that is functionally relevant? So at this point, we're going to switch to the idea of spinal stimulation in humans. And I've done some of the work you're going to see was done by a graduate student in Atlanta when I was still at Emory, but the analysis has been heavily done by Matthias Kren, who's a colleague now at Methodist Rehab. And so I borrowed some of his slides for the sake of that. He makes really good slides. So this idea that restorative neurology is intervening to work with the residual non-injured neural structures to try to improve functional output. And we've talked about using stimulation to activate the sensory input to the spinal cord, which is still present, even if it may be slightly altered in its anatomy and physiology. And the idea is that stimulation of dorsal root afferents, these large fiber afferents, the lowest threshold ones for depolarizing in the face of external electrical stimulation, can be a way in. And this is probably both direct activation of motor units at certain strengths to get reflex output, but also there's access to all the interneuronal pools that lead to the neuromodulation of the spinal circuitry. Of course, this has been a popular thing, and proof of principle has been shown over the last decade for a variety of different functions with regards to improved physiology, and all focus predominantly on locomotor function and enabling movement training. But my frustration with the field currently is that we have a large list of proof of principle papers and a somewhat overzealous attempt to turn proof of principle into standard of practice without the adequate work having been done in between. In 1945, when penicillin came out, it was reasonable to tell everybody we're going to try penicillin on your infection and see if that'll make it better because you have a fever and you look sick. But at some point we figured out that there's different bugs and we need different antibiotics. And if you went to a doctor now with the urinary tract infection symptoms and they didn't culture and figure out sensitivities to your medications, you consider it sort of malpractice if they just handed you penicillin. So things have to progress from proof of principle, we know it can work, to really figuring out how much it can work, in whom, and what's the best way to get it, and really develop a modern methodology here that would parallel what we do for urinary tract infections between 1945 and 2015. So we have focused largely on using transcutaneous stimulation to set up an electric field that's highly focused at the dorsal root entry zones or at the neuroforamina, that's where the sensory axons are, and as they enter the spinal cord. The rationale we've been using for this is largely that it's non-invasive in the sense that we can do many patients very quickly over time for research, which is to say that it's great for being able to study things like stimulation strength and frequency and that sort of thing. It does not have the spatial resolution of intrathecal electrodes, which are shown here. And I suppose people there are familiar with this technology. The upside here might be that there is, a spatial orientation of which electrodes are used and where they're placed on the spinal cord to optimize certain functions. But even with this technology, we've not fully examined the right stimulation parameters across individuals to start learning some general rules about what profiles go with what kind of stimulation parameters for what kind of effects. And of course, since these have to be implanted, they're an expensive process, which makes doing research much more difficult, both time-wise and cost-wise. But I would argue that if we found somebody in this scenario that had a great response and we didn't want to have them put electrodes on every day, this would be a wonderful solution. I'd also make a little bit of a pitch for it's entirely possible that we should do, like we do for intrathecal baclofen or for pain stimulators, some sort of trial. And you could argue before implanting this surgically, you could test a patient with transcutaneous stimulation. And if you got a great effect and could figure out optimal stimulation parameters, then you might be more justified in implanting this rather than implanting it just to see if it works. And we all are familiar with individuals who have had this implantation who have then later shown up somewhere and said, this isn't really working. And either I want it out or turn it off or it's not doing anything. So I think these two things have to be used complementary. I think this will end up being the clinical solution, but this may be the better arrangement for research studies, at least to be able to get through a larger number of patients in a less invasive, less expensive way. So if we stimulate this collection of dorsal roots at a high enough stimulation, we get the direct afferent to motor neuron connections and get these reflexes that are again quantifiable and measurable both in their size and their latency. And they're not exactly symmetric in every individual, whether injured or uninjured. They're more asymmetric in the injured because there's more heterogeneity in the circuitry in the spinal cord and spinal cord injury. But you can map out the relationship between stimulus intensity and the size of these reflexes. These are essentially M waves. These are essentially H waves. There are no M waves that we can detect. If you start stimulating its strengths enough to activate ventral roots, you tend to get a jump in a shorter latency in addition to the waveform, but that's often difficult to sort out. And when I get to our data, I'll show you how we discovered some after the fact that although we were stimulating at stimulation strengths less than would elicit these reflexes, in the heat of battle, sometimes reflexes break through and contaminate what's going on in terms of trying to modulate the circuitry without causing reflexes. So this is some of the most interesting part of this. And I started bugging Dr. Dimitrievich at a Society for Neuroscience meeting when I saw this on his posters that the spinal cord could take tonic stimulation and turn it into phasic output. And that at very low stimulation frequencies, there was these individual reflexes. But as you increase the stimulation frequency, the spinal cord started processing through these spinal circuits this signaling to allow there to be windows when reflexes could be expressed and then diminished, and then allowed to be expressed and then diminished. And it was sort of alternating windows of opportunity between antagonist muscles. And so this really looked very much like the pattern of locomotion. And so it would be very possible to use tonic stimulation to get phasic motor output like locomotion. And so at this point, I was very curious about what might be possible with this technology. I'll admit, when I started off, I thought, well, we know we can do locomotor training in incomplete injuries, but we don't get much out of that with complete injuries. Maybe this will be the thing that will allow us to access complete injury. And it's not been that easy. I'll just say it that way. There's a couple of theories about how this works. The most common one is talked about increasing central excitability. And this is a bit of an overly simplistic explanation for everything. It certainly could be part of things in that you add to, some baseline of excitability that a voluntary command cannot excite, but in combination with spinal cord stimulation and a voluntary command, now you can reach a threshold to generate motor output. The problem is that this doesn't necessarily distinguish between physiological motor output or pathophysiological motor output like spasticity, hypertonicity, clonus. And we can definitely show examples where we are suppressing motor output. And so it's not simply this idea that stimulation is increasing excitability and now allowing everything in the way of a voluntary command to get through. The other thing we talked about was this activation of the neural circuitry that may make up a pattern generator for locomotion in the spinal cord, whether that's a hardwired network or a state that interneurons can achieve. We certainly know enough about reflexes of 1A inhibition and cutaneous reflexes, flexor reflex responses. So there's wiring in the spinal cord to allow for alternate activation, left to right, and flexor and extensor and that sort of thing. So this figure was what made me have a religious conversion, that there was in fact a human CPG, central pattern generator, for locomotion, and that was with messing with both stimulation strength and stimulation frequency. Dr. Dimitrevich could demonstrate in a complete spinal cord injury patient this rhythmic output of the spinal cord. But even in this trace, some of the issues are shown. Getting rhythm out of the spinal cord is not always easy and certainly not accomplishable at all frequencies. But while you see nicely alternating activity in the hamstrings and quadriceps, if you look carefully at the activity in the tibialis anterior and the soleus, while it's rhythmic, it's synchronous. And so the ankle is being stiffened and relaxed more so than flexed, extended, flexed, extended, flexed, extended. So it seems that getting pattern out of complete spinal cord injury is much easier. Getting rhythmicity is much easier than getting appropriate pattern. I've seen many more cases where we have rhythmic activity that isn't necessarily patterned correctly. The other thing to be said is that while this is demonstrable as a possibility, some of Dr. D's postdocs have looked through a large number of records, and the estimate of the number of patients who are clinically complete that can actually convert tonic stimulation into this rhythmic output is, from their report, about 11% of the cases. So while it's possible, it would seem that a majority of cases are somehow not in quite the right physiological state to be able to get exactly this kind of output. So this means that it's not just some easy slam dunk. You put it on everybody and everybody's going to have this nice rhythmic output. It's subtler than that, as is true for most biology. So I want to go back to the fact that we can get more output, and this has often been reported for these mid-range frequencies where a descending command and sensory feedback of, say, stepping is insufficient to activate a motor output. But if you add the neuromodulation, you now can have those two sensory inputs in combination with the stimulation result in a motor output. Likewise, we're able to suppress spastic activity, usually at these higher frequencies. And we see that, well, there's normal or abnormal output that if we add the neuromodulation to it, we can now quiet that output. And the issue with walking is that people with spinal cord injury may walk poorly because they're weak, but they may also walk poorly because they're stiff. And you probably wouldn't apply the same rehabilitation strategy to a very spastic patient that you would to a very weak one. And so the same stimulation isn't probably going to have the same effects in a spectrum of clinical presentation, even though if the only measure of output is gait speed, then you're not necessarily going to know, am I treating the spastic ones or am I treating the weak ones? And of course, in most patients, there's some combination thereof, which makes it even more complex. So I want to tell you a little bit about the work that Jason White did as a graduate student, and that Matthias has done some great work analyzing. What we did was we studied folks in a robotic stepping environment for the purposes of using the robot for measurement. And I'll explain that in a second. But we sort of randomly stimulated at different frequencies throughout a course of 300 steps to see what the overall effect was. And so let me explain a little bit about the setup. So the Locomat is an exoskeleton that uses actuators at the hip and knee to get the limb to the right place at the right time for stepping, given the person's leg length and height and so on. And you can see it looks rather physiological in terms of the motor behavior, how natural the stepping looks. And so we think that the sensory feedback from this is probably somewhat relevant. I mean, there are straps on the limbs, and the person has got body weight, so it's not completely normal, but it's probably close to physiological in terms of the timing and which muscles are being stretched and contracted in which directions. The robot has an assist as needed mode, and so the instructions to the patient are try to match the robot, not try to walk faster or let the robot carry you, but in fact work with the robot to walk in its greatest synchronization between the two, between yourself and the robot as possible. The other thing I would point out is the robot is very good at keeping your leg in exactly the right position at exactly the right time. Now you can control with something called guidance force how precise that is, but at 100% guidance force, this is kept very precise. And the good news from that is every time the robot has to apply some force to change the trajectory to get the limb to the right place at the right time, it's keeping track of that. It's reporting those forces necessary. So this is sort of a real-time ongoing force sensor of what it takes to get the legs to the right position. It may be holding them back. It may be pushing them forward. The leg may be late or early or too strong or too weak for certain movements. But what you can see down here is as the person is stepping, the actual angle that was recorded and the desired angle that was programmed in the device are very, very close. So the device is keeping the leg movement rather good, even with some slop between the cuffs and the robotic arms. And we can measure what those forces look like at the hip and at the knee during that stepping. So it's really the fact that the robot is a recording device that we've used instead of video or relying entirely on electromyography. So here's an example of an individual with no stimulation, 89 hertz, 25 hertz, and then no stimulation again. And you can see that the hip-knee angle is not markedly changed from one condition or the other. This is all being position controlled by the robot. But you can see that the forces being recorded are a bit different under the different stimulation measures. And so we can record how much distance between this force profile and this one occurs and give that effect on force to 89 hertz stimulation. And this force profile relative to this one to 25 hertz stimulation. So we have, here's an example over time of randomly selected stimulation frequencies of epidural, transcutaneous spinal stimulation, not epidural, I'm sorry. And the changes in hip forces during stance and swing and the knee forces during stance and swing. And for this one individual, what we can plot is the relationship between stimulation frequency and the amount of forces that were applied by the robotic device, different from what was going on at baseline. So you can see here in the hip and the knee that during stance with higher stimulation frequencies, the robot had to do more work. And during swing at these higher frequencies, the robot actually did less work. And I'll just wet your whistle a little bit to say, think of this as perhaps this stimulation frequencies, which we've described previously as being able to shut off motor activity, maybe decreasing the extensor ability. And so the robot has to help the person stand more. but during swing, it's gotten rid of stiffness, and so the robot has to help less to get the limb through its movements to get to the phenomenon. So the effect of the stimulation isn't even the same during stance and swing.
So that's what I was going to ask you, Keith. Just to clarify, at baseline or without stimulation, the patient was able to contribute to the gait cycle. When you add stimulation to it, we are further stiffening or further relaxing, or what was your interpretation of this?
Oh, it could be that we're getting rid of tone. It could be that we're augmenting the CPG. It could be that we're just changing the timing. Maybe at some stimulations you speed up or slow down relative to where you're supposed to be in the gait cycle and where the robot wants you to be. So it's very difficult to sort out whether the robot was helping or hindering, or whether it was speeding up or slowing down the limb as to why it had these changes in forces. But the point is that the forces are telling us that something is changing in the motor output of the individual compared to no stimulation, and the nature of those changes.
Okay.
So we studied this in 9 individuals, all shown over here. And what's plotted here is for each joint, the change in force at the different stimulation frequencies, either for stance or for swing. And the colored boxes are the boxes where these changes were significant. So they had to have decent effect size to be counted as significant. There's a couple things that I'd have you notice. And then, well, I'll also say over here on the right, we used the algorithmic clustering process to put like and dislike the different patients, and they sort of sorted into about 4 groups. And this serves a purpose not for saying there's 4 groups within the patient population, but they have some features. So these 4 individuals, the most obvious feature is that during swing, the robot, especially at higher frequencies, caused these blue boxes, which is a decrease in contribution. So that all these values are negative. So the amount the robot had to assist went down. And in these 5 patients, the predominant effect was exactly the opposite. The stimulation caused the robot to have to provide more assistance during swing. The other thing you can see if we look amongst these 2 groups up here is that this group, there's a lot of blue in swing and a fair amount of orange in stance. But in this group, while there's blue in swing, there's not so much orange in stance. So that's sort of what separates these 2, A2 from A1. And if we look down here, there's a less clear of what's going on over in stance. Sometimes it's blue, sometimes it's orange. But clearly these 3 down here have a lot of orange during swing, and these 2 have a bit less. And that's again sort of what is probably the basis for how the algorithm separated out group B1 from group B2. So we're going to propose that there's sort of, at least in this 9 patients, sort of 4 predominant phenotypes. So we then went to look and see, well, what is the effect of stimulation frequency on these 4 different groups? And this is sort of what we see. So the A group, which we know is the mostly adding stimulus during stance and requiring less stimulus during swing, you can see as the stimulation frequency goes up, the robotic forces are progressively increasing. This group, to a great extent, this was the one with lots of blue in swing. This one was with lesser amounts of blue in swing, but they sort of have the same shape. So this is a super responder, and this is a moderate responder in this pattern that says that we're going to do more robotic forces during either increased or decreased at a higher stimulation frequency. So the effect of frequency is to cause the robot to do more of the swing stance dichotomy.
So the higher frequency, the poorer their performance, because the robot is required to help more.
Well, it's required to help or hinder more, because in these 2 patients, if you go back here, they're getting less help during swing and more help during stance. So the effects are, that next figure shows the magnitude of the effect, not the sign of it. So whether it's increase or decrease, but there's more of an effect at these higher frequencies. Look at all these, colored squares here. And there's an effect at higher frequencies in this group, but it's less profound, which is part of the reason why this slope might be less than this one. So if we go back to the maybe decreasing extensor tone and the robot has to help during stance, but that decrease in tone makes it easier to do swing, that might be why these frequencies look like they do. Now at the other end of the spectrum, we have this group of 3 individuals that is sort of what we might consider a bit more classical in that the best stimulation frequencies for changing the forces are the mid-range frequencies, and when you get to higher frequencies, this goes back down. This may be the group where these mid-range frequencies are actually in some way maybe augmenting physiological output, kind of in the classic, we're not using stimulation frequencies to get rid of spasticity, we're not using stimulation frequencies that just are good for tonic activation, but maybe for phasic activation. So this might be the ideal case of where, you know, you could do tuning of the right frequency to get the greatest effect. But this is only 3 out of 9 patients, so a third of the patients do this. These 2 patients didn't have very much change at all in the effect of stimulation frequency. So searching for a best frequency in this group is sort of meaningless. This is probably the most, quote, non-responder group. And if we go back up here, it's these 2 patients here that have the sort of the fewer numbers of squares and less consistency about what's being affected. And sometimes even what's going on at the hip is opposite of what's going on at the knee. So there's even joint-specific kinematic bits going on there. But this certainly would say that not all patients respond to all frequencies in the same way, and that it might be important to know which of these patients that you're dealing with, if in fact there are these 4 groups, and not more and not really less. I mean, you could argue these 2 might be lumped together because they all have the same sort of profile. But looking for the best frequency may not be something that is easily done equally in all patients. And so it would be great to know what makes B1 patient fit better in the B1 category than in the A1 category. And what does that mean for how I might apply neuromodulation? So now this just tells us about forces. We did go look at the EMG, and this is an example of the suppression of muscle activity with increasing frequency. So this is sort of like the A group. So here's the stimulation, no stimulation, and here's the EMG patterns going along here for these leg muscles. And we turn up to 9 hertz and then up to 24 hertz and then up to 50 hertz. This nice pattern that's locomotor appropriate, at least for the proximal muscles. There's sort of co-contraction and clonus down here in the ankle movers. But this pattern that's sort of locomotor appropriate sort of disappears into nothingness as we get up to these higher frequencies. A lot of these stars here are a sign that there's contamination in here of reflexes, of motor responses. So this is no longer sub-motor threshold stimulus strength. At these frequencies, with this person walking, there was some breakthrough reflexes. But you can see that even though there's lots of individual spikes of those reflexes, you know, the envelope of activity is flat compared to here where these nice phasic envelopes of activity. And most importantly, if we go back to at the end of the 300 steps to no stimulation, the EMG patterns are rather similar to what they were at the beginning. So this is an example of sort of getting rid of activity, and maybe getting rid of inappropriate activity would be beneficial, but that's certainly happening here in these ankle muscles. There's much less clonus and there's no more co-contraction. But the extent to which this is helping stepping is a little bit hard to sort out.
And when you look back at the classifications you came up with there, Keith, if you just looked at these patients or examined them clinically, would they look similar to one another? I assume these are all Asia C or Asia D.
So here they all are, and there is absolutely no relationship to C or D, to neurological level of injury, to lower extremity motor score. So there's nothing in this chart. Now, granted, it's only 9 patients. But there's nothing in this chart that jumps out that says C's are more like the A group and D's are more like the B group, or cervical versus thoracic, or the lower extremity motor scores were one thing or another. So we may this is a point we'll get to at the very end, but we may need some additional ways of physiologically profiling people to make any sort of prediction on what they're going to do.
Okay, these patients have relatively good sensation below level, right? Well,
it's, we do very little with the sensory scores on the exams and don't make much relationship between those and the locomotor performance. And, proprioception, I mean, tactile cutaneous sensation and pinprick are not necessarily the afferent information that matters for walking.
Right. I guess my question is it uncomfortable? Is the stimulation uncomfortable in this patient group?
Most of the patients can tolerate up to 80% of the stimulation strength that would activate all the motor pools into reflex activity. But we had some that could only tolerate 20 or 30%. So there's a few exceptions where you can only get away with so much stimulation.
Got it.
but others can handle it. So there's that component as well in terms of determining a good candidate for implantation.
Right.
Again, arguing for why it might be important to do a pre-implantation trial with transcutaneous stimulation.
So your amplitudes may have varied across these patients based on their tolerance.
And their body habitus and
Right.
yeah.
Okay.
So tolerance was one part, but body shape and severity of injury. And in some individuals, the differences in what it takes to activate a motor pool from one different motor pool are relatively low. In the normals, this is about a 20% increase from the ability to get reflexes in the most sensitive motor pool to the least sensitive motor pool. But in some of these patients, it could be much greater percentages. In other words, it may be a stimulation strength that it takes to get any reflex out of the most refractory motor pool. Even 80% of that stimulation strength may be above motor threshold for the least, the most easily recruited motor pool. So it's difficult to know how to describe the stimulation strength relative to the spectrum of physiology. And you can have a quadriceps on one side that's easily activated and a quadriceps on the other side is very hard to activate. And we don't have very good mapping so far for, asymmetry or heterogeneity, but some of these patients can be really kind of wacky in this regard. All right, let me show you in the example of the sort of ideal situation where, there's these motor patterns of alternating thigh muscle, knee activators, and even though there's sort of co-contraction, at least the soleus is active during stance and not so much during swing. And as we increase the frequencies to 33 hertz, these envelopes get larger and perhaps more appropriate and stronger and better stepping. And then at higher frequencies, these things start to go back down. And when we get back to no stimulus, it looks like no stimulus at the beginning. So we haven't permanently changed things, but this is a group that might, benefit in terms of augmenting physiological output as opposed to getting rid of inappropriate physiological output. But I should point out that this increased motor output cannot only be normal physiology. Here's an example of an individual who we actually made worse in the sense of having more pathology. So here's a patient with, nicely alternating flexor extensor activity, but clonus in the soleus and tibialis, more in swing than in stance. But as we start to stimulate, all we really did was markedly increase that amount of clonus, even though we also increased the gait appropriate activity of the more proximal muscles. Again, these are evoked reflexes that aren't part of the activating the CPG with subthreshold stimulation. And this is just an example of things that broke through at the stimulation strength in this muscle in this individual. But it's possible to make pathology worse, at least in part of the nervous system. And even when we get up to 70 hertz, we knock it back down a bit, but not better than baseline. And of course, when we get back to no stimulation, we're back to what things looked like before. So there's this huge spectrum of what can occur. And individuals are going to have different frequency dependent profiles. And that changing stimulation, especially in that group, B2, we cannot always drive output. Although in 1/3 of the patients that we studied, we were able to sort of support this idea that there was a optimal frequency range for augmentation and at higher frequencies, some sort of suppression. Where in, 5 or 4 out of the 9 individuals, we found that the higher the frequency, the more the inhibition. And there was never sort of this peak of a frequency that increased motor output and then decreased it. It just got progressively more and more suppression the higher the stimulation frequency. So this really gets to the point of, because there's this high variability of outcomes and movement patterns, it's important to know in whom and how we apply this electrical neuromodulation, which means we need better physiological profiling of patients with spinal cord injury. This may turn out to be an electrophysiological profile. Matthias is working on mapping the responsiveness of patient spinal cords to electrical stimulation with reflex testing, and in reflex testing with leg movement instead of the Locomat, the Origo, and trying to figure out if you can find, profiles so you could send a patient to his lab and have him do this non-invasive testing, and he might be able to predict for you what they're going to respond like. But we really need to know who are responders and non-responders instead of just slapping electrodes in anybody who says they want one, because the negativity of the non-responders will, put us in the same place that stem cell research has gotten to. We have a great potential there, but because it didn't hit a home run in the first at bat, nobody wants to sign up for a trial and nobody wants to fund one anymore to study that. And I don't want the same thing to happen in neuromodulation. So being able to predict ahead of time, even if it means a trial with cutaneous stimulation, you might be able to detect who is and who isn't responding and implant fewer of the non-responders and focus more on the responders. And we also need to figure out who's not walking well because they're stiff versus because they're weak. And, which modulation strategy is best going to help that individual walk better. Is it getting rid of spasticity or is it augmenting stepping? So we really will need once we've identified the right patients, we're going to need some sort of standardized algorithm for finding and adjusting optimal stimulation parameters. The location, the frequency, the intensity, whether it should be a continuous thing or whether it should be patterned. Gregoire Cortine has been using afferent input focused at appropriate levels at the appropriate times in the gait cycle, but that's, to my mind, more like stimulating peripheral nerve than really activating the CPG. This is just sort of trying to do for the nervous system what the muscle sensory feedback is doing anyway, just augmenting it, which may be a good solution for stepping, but isn't necessarily engaging the circuitry in the spinal cord. Because if we have to do random testing in each and every individual, we're going to be really frustrated and it's going to take a lot of time. And then, of course, we're doing a really lousy job, in my opinion, of monitoring the nature, the magnitude, and the duration of the benefits of neuromodulation after people are getting stimulated. I don't know how well the patients who have been implanted are being monitored or being tracked as to how long this effect lasts. In computer brain interfaces, they often find they have to recalibrate almost on a daily basis so that the interaction between the external recording and stimulating protocols and the neural axis is fine-tuned. And if we put something in and it works one day of the week or works for 3 hours and then after that doesn't work very much, or you can entrain or exhaust the circuitry, then this may not be of always of the best benefit. So knowing whether you should just flip it on when you want to get up off the couch and walk into the kitchen or whether it should be on all the time remains to be seen. But we have now a fair number of people implanted, but the large numbers of the centers that are doing most of the implanting don't seem to be doing a huge amount of post-operative monitoring about what the effects are, bringing people back in and retesting, say, their reflexes or their EMG or their forces in a robot or anything like that. So I think it's early days. I think we have great proof of principle. I don't think we're anywhere close to standard of care because there's too much we don't know and a lot more we need to find out. And I think some of this requires that we think a little bit more open-mindedly about what might be going on here. We've not considered much about the effect of stimulation on those residual ascending pathways that are then likely impacting the descending voluntary commands. We've not thought too much about the sensory plasticity that might be going on after the injury and that it may not be homogeneous across patients. There may be some with a lot of afferent sprouting. There may be interactions between pain and non-pain afferent inputs. So there's a lot more to learn, and it's an exciting time in regards to how much more to learn. But I think that the tendency for us in this field to jump from proof of principle to standard of practice sort of ends up being a stick in our own eye because we didn't know what we didn't know, and we weren't willing to think about it critically, and end up with mixed outcomes and not being able to say who's going to do well and who wasn't. So hopefully we'll get to the point where we have a better ability to carry out this kind of work. So I think I've spoken for my time, and thank you for your attention. I'd be happy to answer any questions.
Thank you very much, Keith. That was excellent. And, we hear from different folks very widely varying views on what you can accomplish with stimulation, and yours seems to be very level-headed. Do you at this point in time think that you can identify a patient, or is there a subpopulation of patients that you think would have these miraculous responses to this that have hit the news and that people have been cheerleading about? Do you think that's just a red herring, or what are your thoughts?
No, well, I suspect that there is a profile, and I suspect the people who've been doing a lot of this are better at recognizing it. I think that we have seen a bunch of, well, clinically complete who are you know, a few centimeters away from being incomplete, and they're clearly discomplete. And so we turn a paralyzed person into someone who can stand or move their toe with the spinal cord stimulation, gets a lot of you know, excitement, but it's not exactly fixing paralysis. It's just going from subclinical to clinical incompleteness and effect. I also suspect, and I've actually read grants that have claimed that they do cherry picking. They somehow have figured out who it is that's most likely to respond. But we've had at least 5 years, if not more, of recurring proof of principle, where the salesmanship that goes on with having picked, you know, the best candidate without actually being able to objectively identify for everybody else what's the best candidate has made it kind of difficult. And I don't know whether it's clinical intuition or, you know, some test that's not being shared with the field. But I mean, if you look at our INSCI exam, we don't even make reflex as part of the standard testing, and use of a reflex hammer is woefully inadequate in, you know, characterizing these patients. When I ran a spinal cord injury consult service, I used to offer a free steak dinner if you could find a neurosurgical intern with a reflex hammer in his pocket, and I never had to pay up. And you know, most physiatrists look at me like I'm crazy for taking my reflex hammer to a spinal cord injury clinic as my only tool, because the patient can't lie about the reflexes. They can not try with strength testing, and they can tell me whatever story they want with sensory testing, but the reflexes tell me something. So it may be that there's some profile that if somebody is really hyperreflexic or hypertonic or spastic in a certain way or a certain pattern, maybe that predicts, you know, the A group more than being in the B group. But even polyelectromyography to figure out how much co-contraction and dyssynergy is going on may be helpful. If someone's got a very small amplitude but very well patterned EMG, then maybe you just want to strengthen that. Somebody's got great motor output in terms of magnitude, but it's all disorganized and all co-contracting and dyssynergic. Maybe the goal is to reorganize the pattern. And I think the therapists sort of can recognize the difference between these two people, and they provide different therapeutic attempts to modulate that. But I don't think we can tell, you know, by an exam at this point, you know, which patient is which and what to do with them, especially when it comes to neuromodulation.
Do you think that at this point you could recommend an approach if somebody came in and they said, hey, I want to find out if I'm a responder to stimulation? That you would say, here's what we would put them through to identify that?
Yeah, I think I would tell them that, you know, we don't know enough to know how much or how long or how good of a responder they particularly are going to be. But that in light of the fact that, you know, this is sort of like telling the person with a fever in 1945, we don't know what penicillin works on, but it might work on you, so let's give it a try. So I would take that person, I would do transcutaneous spinal cord stimulation. I do it in a setting where I could make measurements, perhaps other than a video, at least some electromyography, maybe even recording the forces in the locomat to get a sense of what their response is, whether it's frequency dependent and whether it's actually, you know, helping their gait or not. And then, you know, with that in hand, decide whether to implant and what stimulation parameters to set, at least at the time of implantation, and some plan for very close following and tweaking and adjusting of stimulation parameters to get the required output. And it may very well be that the best stimulation parameters for walking are not the best ones for standing, are not the best ones for autonomic dysfunction, or not, you know. So, you know, in any one individual, they may end up with having 3 or 4 programs. I flip program 1 when I'm feeling like I'm getting a bout of autonomic dysreflexia. I flip number 2 when I want to get up and, you know, walk across the room. And I flip number 3 if I just want to, you know, stand up well. And 4 if I want to be able to voluntarily do more with my fingers or toes. And, you know, the cervical injury is an entirely additional difficulty because there you've lost sensory input, motor output, as well as supraspinal input. And so the heterogeneity in cervical spinal cord stimulation is going to be 1,000-fold worse and more difficult to approach than looking at the lumbar enlargement when the injury is remote from that, thoracic or cervical.
Thank you very much. Dr. D, I see you on there.
You're muted, Dr. D.
You had to unmute him. We're going to unmute him. Right there, S to unmute. Did you do it?
We can't hear you, Dr. D. You're muted.
Dr. D, you've got your mute on, so we can't hear you yet.
Still can't hear you.
Did you see the microphone with the red slash through it? If you can click on that, it'll turn it on so we can hear you.
It's at the bottom of the screen.
Bottom of the screen, far left. All the way to the left at the bottom. And one other question for you while we're waiting for him to connect up. Was the profile you're seeing when you bring them back in the future, if you go back to that same stimulation setting, are you getting the exact same response every time? Is it a reliable response?
So we only studied 3 people in more than one setting, and there was a varying degree of variability. But this is an incredibly important question if you're going to implant a chronic device in someone and count on it working the same way every day or to work the same way over time. Because it's entirely possible that this change in activity will change the neural circuitry there, and what will be effective early on may not be effective a week, a month, a year later, or vice versa. And you know, there's at least a subset of people with intrathecal baclofen pumps who have had them removed later because they didn't need them anymore. Yeah. And who knows if that's what we'll experience when we get to the point of implanting these people. But we've not, because this isn't an implanted device, we've not looked at people 6 months later or a year later or anything like that, or before or after training. This has all been assessing the circuitry that's present in the spinal cord at the time. We've tried to study people who haven't gotten a lot of locomotor training in attempts to really understand what's going on with the injury profile that's left, rather than the effect of that plus some sort of intensive rehabilitation. But there are those who swear that this only works with rehabilitation, but nobody's been willing to do the study where they do, you know, an arm with stimulation, an arm with therapy, and an arm with both, and an arm with neither. Because the motivation is to sell the power of this new technology as opposed to really understanding in whom what works.
Yep. All right, I think Dr. D's got his microphone working.
1, 2, 3, can you hear me?
Yes, now we can hear you.
Thank you.
Again, my message is following. If you like to restore what you lost, it's not only to assess deficit, it's also to understand the underlying mechanism of deficit if you like to modify the deficit. I like very much, Kate, what you said, that you're coming with the reflex tools, and we are in the time when we are going to change our neurological protocols for assessment of potential to modify the output and sensory activity. Let's continue. Beautiful.
Thank you very much, Dr. D.
Perfect. We better run to clinic. Thank you very much, Keith. That was excellent. Appreciate it.
It was a pleasure. Good talking with everybody. See you next time.
Sounds good.
Okay. Bye-bye.
I think it stopped.